Welcome to our channel Mootechie, exploring the invisible world. This is where we begin our journey into the unseen. Now, let's get started with an introduction to microbial classification. First, we look at organizing diversity. This is the process of grouping the vast microbial world into meaningful categories based on shared traits. Think of it as creating a filing system for millions of different microbes. The field has evolved from phenotype to genomics. We've moved from just observing physical features to analyzing genetic blueprints and evolutionary relationships. This work is of critical importance. It forms the foundation for medicine, diagnosis, biotechnology, and ecological studies. Without it, science would be navigating in the dark. Let's explore the meaning of microbial classification in more detail. At its heart is the taxonomy core. This is the science itself, which comprises three distinct but interrelated parts: classification, nomenclature, and identification. It's the entire process of naming and organizing life. This leads to systematic grouping. It involves organizing microorganisms into groups based on their shared characteristics and evolutionary relationships. We put like with like based on science, not just guesswork. This structure is formalized in the taxonomic hierarchy. This is a ranked system that flows from broad to specific, starting with domain, then kingdom, phylum, and so on, all the way down to genus and species. Each step down gets more and more specific. The ultimate goal of all this work is to create a stable, predictable, and universally accepted system for studying microbial life, a common language and framework that all scientists can use. So, what is the need and importance of classification? Why do we bother? First, it provides a universal language. It enables precise communication across the global scientific community, ensuring every researcher speaks the same language. A bacterium called E. coli means the same thing in labs from Tokyo to Toronto. It's essential for diagnosis and therapy. Accurate clinical diagnosis and choosing effective treatments depends on it. For example, determining if a bacterium is gram-positive or gram-negative directly guides which antibiotic a doctor should prescribe. It forms the foundation for research and biotech. Classification drives innovation in biotechnology, industrial microbiology, and advanced research studies. Knowing exactly what you're working with is the first step to any application. And it's crucial for ecology and public health. We need it to understand microbial roles in natural ecosystems and for tracking disease outbreaks in public health surveillance systems. It helps us monitor the health of our planet and our populations. Now, let's walk through the history of microbial classification, a journey from discovery to genomics. In the 1570s, it began with discovery. Antony van Leeuwenhoek first observed animalcules using his simple microscopes, revealing a whole new world. Later, scientists focused on morphology. Ferdinand Kohn took a major step by classifying bacteria primarily based on their shape and form, like rods, spheres, and spirals. A big shift came in 1969 with the five-kingdom system. R.H. Whittaker proposed the five-kingdom classification, which included Monera, Protista, fungi, plantae, and Animalia, grouping life by cellular organization and nutrition. Then, in the 1970s, a revolutionary concept emerged: the three domains. Carl Woese and George Fox used ribosomal rRNA or rRNA sequencing to establish the three domains of life: bacteria, archaea, and eukarya. This showed that archaea were fundamentally different from bacteria. And today, in the 2000s and beyond, we are in the age of genomics. Modern taxonomy uses whole genome sequencing or WGS and average nucleotide identity or ANI for incredibly precise identification, down to the very letter of an organism's genetic code. What is the basis of classification? What methods do we use to categorize and identify microorganisms? We start with phenotypic characteristics. This is based on observable traits like morphology, which is its shape, staining reactions like the famous Gram stain, and whether it can move, its motility. It's what we can see under a microscope or test in a lab. We also use biochemical properties. This analyzes an organism's enzymatic activities, what substrates it can use, and the unique metabolic pathways it possesses. Different species have different biochemical fingerprints. Another way is through ecological criteria. Here, we group organisms by their habitat, their environmental roles, any symbiotic relationships they form, and whether they cause disease, their pathogenicity. It's about where and how they live. The modern standard, however, is genotypic and phylogenetic analysis. This involves sequencing genes like the 16S rRNA gene or even whole genomes to trace evolutionary ancestry. It's the most powerful tool for seeing the true family tree of life. Because each method has strengths, the best practice today is a polyphasic approach. Modern taxonomy combines all these methods, phenotypic, genotypic, and phylogenetic, for the most accurate and robust classification possible. Let's summarize the key advantages of classification. It brings consistency and clarity. By ensuring universal naming standards, it eliminates the confusion caused by regional or common names. A rose by any other name might smell as sweet, but a pathogen needs one exact name. It provides predictive insights. Once an organism is classified, scientists often can predict other traits, metabolic capabilities, and even behaviors based on the known characteristics of its group. It turns a single discovery into a source of information. It's indispensable for clinical and public health, from accurate diagnosis and targeted treatments to effectively tracking and containing disease outbreaks. A good classification system is a frontline public health tool, and it enables advanced research and the study of biodiversity. It facilitates global studies of life's variety and allows seamless data integration and comparison across research labs worldwide. It helps us piece together the grand puzzle of life on Earth. Thank you. Subscribe to Mootechie for more. We hope this exploration has helped illuminate the fascinating and essential science of organizing the invisible world around us. Keep wondering, keep exploring.